So everyone wants to meet an alien, but we're not talking about little green humanoids today. I know it turned off. Yeah, I don't know why it turned off. Just working in. Let's see. So we are talking about like maybe simple single-celled bacteria. We don't know. And I'm here to tell you about 3 frontiers in this whole search for signs of life beyond Earth. I'm going to need our expert IT professor to help out, but I'll start talking anyway. So this story actually begins billions of years ago, when something incredible happened here on our Earth. Simple single-celled bacteria figured out how to harness energy from our environment. You're all in the biology department, most of you, so you know exactly what I'm talking about. Photosynthesis. This was such a remarkable development on Earth's time. And these cyanobacteria figured out how to get energy from our sun, and in the process, generated oxygen, which was a waste gas for these bacteria. But this is super important for the astronomical search for life beyond Earth, because oxygen is highly reactive. It should not be here in our atmosphere. It is so reactive, it should react away. And indeed it did initially. It took a long time for oxygen to accumulate. But we now hold oxygen dear in our hearts as a way to find a sign of life beyond Earth. And what's so cool about all this is it was 100 years ago, Jeans 1930, this astronomer wrote about it in a textbook. And he said, like what it says there, he just said, wow, like our vegetation is continuously discharging oxygen. Without a continual production of oxygen, it would just go away, it would react away with the surface and get destroyed by other gases and photochemistry and things like that. So that's kind of the basis for the search for signs of life on exoplanets. And I do like to imagine that there is a planet orbiting a nearby star, because all stars are suns. And there's an intelligent civilization there with the kind of telescopes we're about to build. And if they're looking at Earth, they're going to suspect there's life here. So it's not from big things like the Great Wall of China, it's not from pollution or city lights, but it's going to be from a gas that is way out of equilibrium with an environment that doesn't belong. On the other hand, these aliens, if you work in science, they're probably fighting over data analysis. Some might be saying, terrible place for life, oxygen's too reactive. It's gonna have large regional fires and there could never be life. So we don't really know, and it is a bit of a long journey. So what I'm gonna talk about today was, I'm going to tell you a bit about exoplanets, and then I'm gonna veer off onto our sister planet, our so-called evil twin sister Venus. And then I'll launch into another topic, ionic liquids, and I'll show you how they are all connected. I just put this up here in case someone wandered in who is not a scientist or a science student, but it's just first to remind you that every star is a sun. And we astronomers have found thousands of planets, and we think that there are many planets around each star, meaning in our Milky Way galaxy alone, there are trillions of planets. But the point for you that is relevant for today is that although we have found thousands of planets, did you know there's no solar system copies yet known? And that isn't because they aren't out there, it's just because they're hard to find. Like we have our sun, then Mercury, Venus, Earth, Mars. Relatively tiny low-mass planets compared to our sun. Then we have Jupiter, Saturn, Uranus, Neptune. We don't even think giant planets are all that common, if you want to know the honest truth. And it's just for all of it would take me an entire semester class to explain to you why that is. But like a selection bias enabled us to find other types of systems with big planets close to the star or small planets close to the star. We can't find solar systems. But what this has done for us is it sent us off like on a tangent of trying to open our minds and not be so Terra-centric, so fixated on Earth and Earth processes. Because other planets are just so different from our own. So what I decided to tell you out of all the millions of things going on is just how we're trying to find signs of life on an exoplanet. So I'm going to walk you through this, and it's a bit tutorial-like, so just bear with me. But out of all those planets out there, there's a special subset that are fortuitously aligned. They happen to go in front of their star as seen from our viewpoint. Because all orbits are random. So a planet could be orbiting in the plane of the sky, right? And we wouldn't see it go in front of the star. It has to be like just so. So there's a subset, a small fraction that will just be randomly, perfectly aligned from our viewpoint. Because we don't spatially resolve a star at all. A star is a point of light, and when the planet goes in front of the star, as seen from our viewpoint, it drops the starlight by a tiny amount. It's related to the planet area compared to the star area. And this, for a giant planet, it's about 1%. For an Earth-sized planet, it's about 1 part in 10,000. So you can see this real data here, taken by the James Webb Space Telescope. It's showing you the relative brightness of the star. And in astronomy, by the way, we just like to normalize everything. So we call the star like 1, or it's 100% bright. And then it's dropping in brightness when the planet goes in front of the star. And then the star returns to its normal brightness. So I don't know if any of you have heard of this. I'm just trying to gauge how much people know about exoplanets. Okay, some of you. Okay, but in the whole, what we do is we have a space mission called TESS. There's large ground-based telescopes. We look at tens of thousands to millions of stars at one time. We get our data down to Earth. We use computers to look for a characteristic signal. But the computer finds a whole bunch of stuff, kind of narrows it down to say a few hundred objects out of, say, 30,000. And then we have like humans and other algorithms to go through and line up the ones that are planet candidates. And then we farm those out like to the astronomy community, and they follow it up. It's a very long process. But what I wanted to talk to you about today was how we study atmospheres right now with these transiting planets. And you can see on top, there's a very exaggerated planet with a very exaggerated atmosphere. So the planet's a certain size, but if the planet has an atmosphere, it's a tiny bit bigger. And this whole method relies on the fact that all gases have a spectrum. Each gas supposedly, it's not strictly true, but has like a unique fingerprint of where they absorb light. So this is a spectrum of our sun, like a rainbow broken into a lot of pieces. And all those lines are like gases, mostly in our sun's atmosphere, but some are in Earth's atmosphere. Most of them are atoms, actually. And some expert would know like what each and every one of those is actually. So we're just using this phenomena that different gases absorb at different wavelengths. So I want you to imagine a wavelength where the atmosphere is transparent and the planet is a certain size. So it's going to have a drop in brightness of some amount. Now I want you to imagine another wavelength where the atmosphere is very strongly absorbing. Now the planet's going to look a tiny bit bigger. Are you with me? So we're looking for a change in the drop in brightness as a function of wavelength. We're going to try it. Here is a giant planet transit light curve from the James Webb Space Telescope. So the star is constant in time, it's normalized to 1. It drops in brightness when a planet goes in front of the star, and after it's done, it returns to its brightness. These are different wavelengths. So some wavelengths are transparent, the planet's a certain size. At other wavelengths, there's a very strongly absorbing gas, and the planet looks a little bit bigger. The drop in brightness will be more. You can see the wavelength on the top right, it's in infrared. So your job, it says here, is at what wavelength? Well, first of all, do you see a drop in brightness that's bigger than the rest? And this curve is like an average. The curve is an average, and you're looking at wavelengths, different wavelengths. So someone can now shout out like where you see it. Yeah, I heard a 4.3, a 4.2. Yes. Wow. Let's just let that land. By eye, you have seen evidence for an exoplanet atmosphere. That's awesome. Great job. And what we do, if this was like a class or you were a student, you wouldn't just be kind of obviously doing it by eye, but you would be using computer code to fit this light curve and see actually what is the difference with wavelength. And that's what this next spot is. Now we're looking at amount of light blocked, and we're looking at wavelength of light. So you said around 4.3, good eye. So basically the planet is about the same size until this wavelength. It's very strongly absorbing, and now the planet looks a tiny bit bigger. And it's like, colloquially like a skunk spray. You only need a tiny amount to make a huge difference. And what that gas is it's actually carbon dioxide. Carbon dioxide is very, very strongly absorbing. You know that on Earth, it's one of our most significant greenhouse gases. But I just want you to know that, wow, we can study exoplanet atmospheres. And the same planet at much broader wavelength coverage, look at all that. Planet looks bigger at some wavelengths than others, and you can piece it all together using computer codes, and quantum mechanics tells you what molecule is absorbing where. So I just wanted to introduce that way that we study atmospheres. We're pumped as a community because we're the first generation that has this kind of data, that can look at exoplanet atmospheres. And we're the first ones that can actually try to look for signs of life. It sounds good, right? Well, we ran into some really huge problems. And that's why I'm going to shift gears and talk about a different topic. But first of all, one of the issues is we can't use this for Earth-like planets. And that's because our sun is so big compared to our Earth. And here's a real picture of our sun, here's a fake Earth. Look how big that is. Remember, it's area ratio. That's not happening right now. But there are lots of other stars out there, and the most common type of star are these small red dwarf stars. And so we're studying small planets with atmospheres around small stars, only it kind of appears that the small planets around small stars don't seem to have atmospheres. And this has to do with the star and the star's history. And there's people actively working on this topic, so I encourage you to invite them to give a talk if you want to hear more about like the current status of what's going on and all the nitty-gritty details. So that's Professor Sukrit Ranjan in LPL. So that's that, but I wanted to also just give you a little bit of dirty laundry from our field and explain to you why it's really so hard, actually. It's partly because we don't know if these small planets that are accessible have atmospheres, but it's a little really messy in our data techniques. And I know that depending on your field, your signal to noise can be high or low, but I think it's fair that like at every field at the very frontier, you have low signal to noise. I think that's maybe a truth right there. So I'll show you what is going on by this cartoon, and in your head you can hopefully put the analogy to your own work to understand it. So we have a transit far left, we take some data, we get this spectrum, which is the size of the planet as a function of wavelength, we fit our data, and we have this method, it's an umbrella term called retrieval, but it's how do you get from the data to the physical parameters you care about? How do we get from our data to what gas is and how much of it gas there is? So we call that retrieval. So in that little cartoon, we're just showing some probability that we have gas A or gas B. In the best case scenario, yay, it's a sign of life, that would be awesome. But in reality, of course, it's messy. Our star has spots, okay? It has flares. It's not pure and clean. And those things are evolving with time. So that's a problem. So now in this cartoon, it's not as a clean picture, we've got noise. That means when we're trying to get our data, like our spectrum as a function, or size of the planet as a function of time, our error bars are bigger. Moment, just assume, fast forward, that we've solved all our problems, that we can look for signs of life. What are we gonna look for? Well, oxygen has only been significant in our atmosphere for about 1 billion years. Sounds like a long time, but it's maybe 1/5 of Earth's life. So what about those other 4 billion years? What if there's other planets that haven't had the chance? What other gases would we look for? Well, you know there are thousands of gases produced by life. Like, you know, isoprene, there's all these gases, the VOCs, but there's like methane, nitrous oxide, like mold in your fridge, like go to the wetlands. So many smells, like, you know, in the desert coming off the bushes. There's untold numbers of molecules. And I did set out to literally categorize every single molecule that can be volatile at room temperature and pressure. And that's another story of what you might call chemoinformatics. But nonetheless, it was to prepare for this. Like, what if we see a signal and it's way out of context with this environment? We want to make sure we don't miss like any sign of life. Long story short, didn't need all that machinery to come across what is pretty much my favorite biosignature gas called phosphine. Now, we don't have a lot of phosphine on Earth here because phosphorus goes with oxygen and phosphates. But there is phosphine, and we don't know the exact biosynthetic pathway to make it. So biologists don't always like this part of the talk. But phosphine, wow, it's very rare, but on Earth it is only associated with life. That's pesticides that we humans make. It is life in oxygen-free environments like wetlands. And it's why do I love it as a sign of life beyond Earth? Because it has like a unique spectral feature. It's not cluttered by like carbon dioxide or a host of other gases. And the fact that it's really, really hard to make phosphine on Earth, like on our temperature and pressure, it's what we call thermodynamically disfavored. So I was like doing this hypothetical computer modeling and theoretical exercises with my team, when wow, lo and behold, an astronomer across the globe, Professor Jane Greaves, she also came across phosphine as an gas of interest. And she herself wanted to find signs of life on Venus. And this is an incredibly way out there topic. And one thing that we love to do in science is just take a small part of our time, and I won't say blow it on something risky, but it's like a splurge. And in fact, right before our meeting here, there was a talk, it's a general ed for undergrads, called Personal Financial Foundation. But I don't think in that class they're saying take some money and blow it. But it's kind of like that. So she was doing that. Professor Jane Greaves wanted to search for signs of life on Venus, and we'll get to why that's crazy in a moment. And she decided to look for phosphine because its ground to first excited rotational state is in the radio, and she's a radio astronomer. So someone connected our two teams, because it's very, very obscure for astronomers to be thinking of phosphine as a sign of life. So we agreed to join her team, spent a bunch of time on this, reported the detection of phosphine in the Venus atmosphere, and the entire finding was trashed by the community. And there were a lot of issues. Just gonna kind of run through now, like the kind of debrief. One legit question, of course, is the signal real? Okay, that's a pretty serious one, but all data is public in astronomy because we share large facilities, and some astronomers looked at the data, some didn't recover the signal. It's a tiny, tiny signal in a giant noisy data set. Some did recover the signal. So we're gonna now assume for a moment the signal is real. We're gonna move to point 2. Then you wanna know, is the molecule correctly identified? Is it phosphine? Out in the radio, signals are really spread out, molecules are really spread out, but someone's like, well, maybe it's something else, maybe it's sulfur dioxide, a gas that's known to be on Venus. We have arguments as to why it would be phosphine. Let's assume the first two are right. Is the signal real? Is the molecule correctly identified? Then we have to ask ourselves, well, is the molecule made by life, or is it made by an abiotic false positive? Is it made by some non-life process? Well, I'll tell you what, it's true that it shouldn't be quantity over quality, and we did quantity and quality. We wrote like 100 pages going through all the known chemistry on Venus and how phosphine could be produced, like lightning, meteoritic delivery, volcanoes, lots of things. And we could show that in every case, using reasonable parameters that we think exist on Venus, you couldn't produce phosphine in a high enough quantity to match the reported observation, right? But nonetheless, it's still, we don't know what happened on Venus. It's extremely challenging. And I just wanted to leave this thought with you, not to have a negative ending to the search for signs of life on exoplanets, but just a kind of reality check about how hard it is to understand chemistry on a planet near or far. All right, so to summarize this part of the talk, Frontiers 1. It's the first time we have the capability to try to find signs of life beyond Earth using telescopes and exoplanets and gases. There are a lot of gases that could qualify as biosignature gases, not just oxygen and methane and the big gases we think about here on Earth. It's been challenging, but we're at the frontier and there's a lot of work ahead. So, all of this work leads to Venus, because Venus not only just has this very controversial report of phosphine, but Venus has so many anomalous gases that have been reported over the years. And what this whole phosphine thing did was, it's so controversial, but it shone a new light on Venus. Phosphine was compelling enough to revive this idea that maybe there's life in the clouds of Venus. And we then, and others, went back and looked at all the things, and I don't have time to go through them all, but oxygen, for example, shouldn't be on Venus, but it's there in part per million, tens of parts per million. And this was found by different spacecraft that went down through the atmosphere of Venus decades past, by different spacecraft, different countries, former Soviet Union and USA. There's all kinds of crazy things on Venus. And you can't necessarily say, okay, a bunch of anomalous gases equals life. You don't say that, but there's a lot of intrigue here on this planet. So there's that. So why is it crazy to think of signs of life, Venus being habitable? Well, you all probably learned at some point that due to a massive carbon dioxide greenhouse atmosphere, Venus's surface temperature is extremely hot. It is 700 Kelvin. It is hot enough to melt lead. You can't even imagine going to the surface. It's just some of the spacecraft landed actually and survived for an hour and sent back pictures, but nothing can really, nothing we know, no kind of life we know could exist there. But just like on Earth, if you hike up a mountain or go in an airplane, it gets colder and colder, and that's also true on Venus. And so that's why half a century ago, Carl Sagan first put out the idea that maybe there's life in the region in the clouds where it's the same temperature as here on Earth. And not as important, but interestingly, it's also the same pressure there as it is on the surface, so it's kind of weird. But at the time, they didn't know what the clouds of Venus were made of. And they're not made of water. They are made of concentrated sulfuric acid, ranging from, we think, like 98% weight by weight down to maybe 80, 70%. So seriously terrible, terrible, caustic, toxic liquid. But here on our Earth, we have life in the clouds. The bacteria are temporarily swept up and they get rained out. And so maybe there's still something there. Now, unlike on Earth, where we have, well, you have no clouds here, right? But we have fragmented clouds. Venus has clouds not from convection, but from photochemistry, and they're always there. They cover the entire planet. They're like super deep. And so maybe there's something about it. And this acid is nasty. Like anyone here ever worked with sulfuric acid? Well, yeah, a good number of people. Usually use it to clean things, or like destroy life. So, we can't really blame people for thinking that you can't have life in the clouds of Venus. You really can't. And just in case you aren't familiar with sulfuric acid, I've repeated this canonical experiment where we're not going to put life in sulfuric acid, but instead of life, we could think of DNA, and instead of DNA, we can think of sugar. So it's like the sugar phosphate backbone. So there's this experiment, you can find it all over YouTube, where people are doing this. But if you look on YouTube, they're not wearing the same gloves I'm wearing. They're wearing gloves that I don't think are resistant to sulfuric acid. So we're going to see what happens when we pour sulfuric acid onto sugar. This is just table sugar, like you would have at your apartment. That's some sulfuric acid. This video is sped up 2.5 times, so you can see what happens. And remember, this is the proxy for life in sulfuric acid. On the right, you can see the temperature from an infrared camera. It's not looking good, right? Like, no, right away something bad's happening here. And it's quickly heating up. It's an incredibly exothermic dehydration reaction. And look at it. That's awful. That is water, actually. It's like water vapor coming off, and there's a secondary reaction that puts off sulfur dioxide. That's nasty. So I won't blame a single person for thinking it's absolutely insane to consider life in Venus. But, part of science and part of life is taking risks. So I don't know. I just don't know what to say about this. It's a fun experiment, and we don't let people touch the outside. We have a theoretical physicist, because I'm trying to figure out ab initio quantum mechanics, like, molecular dynamics as well in the lab. So we had one person visit who worked in that area, and we used to let people touch the outside to see how hot it was. And he's like, ow, my finger hurts. And somehow this theoretical physicist, he got a bit of sulfuric acid on his finger. I don't know how. So it's bad. And one guest wanted to smell it. No. But it actually smells like cotton candy, because it's just like dehydrated carbon, but with maybe a bit of sugar residue. So just FYI on all that. So we're not going to let this stop us. It sounds really insane about that, but we aren't. And there's a longer, personal story of how I moved into this field, but I'm just going to tell it to you scientifically. So we could do a very simple experiment and take our 20 biogenic amino acids and put them in sulfuric acid and see what happens. And these amino acids, I don't have to tell you, they're very safe. You buy like a white powder, you put it in the sulfuric acid, it dissolves. Sulfuric acid being very polar, by the way, it dissolves a lot of things way more easily than water. Put these in, they're all fully protonated, and guess what happened? They're largely stable. And the way that we did our analysis, we used NMR, nuclear magnetic resonance. So what you're seeing here is alanine. We put it in sulfuric acid, like in a tube, and take it to the NMR. And what you're seeing here are the 3 carbon peaks. When I give this talk to astronomers, I have to explain how carbon is not shown with a C on the molecule. But anyhow, you can see where the numbers correspond to. As I'm not going to explain NMR. I'm going to assume everyone here knows what these peaks mean. But even if you don't, what you're supposed to look at is there are actually 3, each of these peaks has 3 different things going on. We've overlaid this amino acid in concentrated sulfuric acid, 98% weight by weight, the rest water. 81%, that's to reflect the different amounts that we think are in the clouds. And then red is just water. But you can't really see a difference because they overlap. And that means the structure of the amino acid is the same in each of these solvents, in water, in pure concentrated sulfuric acid, and in sulfuric acid with a bit of water. So we did this, and it was super exciting. Here you can see tryptophan is the only one that breaks down, but it could have some other problems going on with oxygen, so we're not sure. Some of these amino acids, they get chemically modified, they're sulfonated or sulfated, but the takeaway is that the backbone of the amino acid is stable in concentrated sulfuric acid. So you're gonna ask me, what about peptides? And then we kind of made a bit of a mistake and we went overboard studying the dipeptides. And they are largely unstable. I think a lot of you know about acid catalyzed hydrolysis. If you have some water, they're just gonna break, acid in the water, they're just gonna break apart. But we did find something interesting. So I'm gonna pose a question to you, which you can try to answer whether or not you know anything about organic chemistry. So we took now glycine and alanine and we put them together in all the ways they can. So you have alanine, alanine, glycine, glycine, glycoalanine, glycine. And if you look at this, like it's not always true, but in this case it is. If it turns color, it's bad, it got destroyed. So alanine, alanine got destroyed. Glycine, glycine was fine in sulfuric acid. So what do you think happened here? Do you think that Ga versus Ag do the same thing or a different thing? I just want to let you think about it, you don't have to answer it. Well, lo and behold, one was stable and one was unstable. So we didn't know what to expect here, but in our NMR we found that in concentrated sulfuric acid, it's not having acid catalyzed hydrolysis. It's breaking off in a totally different way because our end product is not two identical amino acids or two of the amino acids. And here I just showed where the bond is breaking for your acid catalyzed hydrolysis versus this new method. But what happened, I can't explain it to you because we don't know exactly what's happening, but we know it has to do with this methyl group. Because, you know, I like to think of sulfuric acid where the hydrogens are coming on and off, like there's fast exchange, and it then kind of exposes this methyl group to further reaction, and it has a cascade of problems which ends up breaking up the molecule. So what we did, in the paper we worked through it in case anyone wants more detail, but we did, instead of CH3, we put a CF3 on here. Because fluorine is like super tightly bonded, and nothing happened, this whole molecule is stable. So the message here is that, wow, chemistry is different in concentrated sulfuric acid, and it's like a huge thing. No one has any intuition. We don't know what's going on. We're trying to work through everything bit by bit, but I just wanted to give you this example of a molecule in concentrated sulfuric acid. And why was this bad? Because we sort of naively thought, like, okay, so anyhow, let me keep moving along. Even though our DNA is unstable, we took our nucleic acid bases and put those in sulfuric acid. And these are all stable. All of these were stable even for more than a year, or more probably, almost too stable. These nucleic acid bases are all stable. But one thing about these nucleic acid bases, I just have a little funny story to tell you. So as an astronomer working in astrobiology, I'm happy that the rings didn't break apart. Like they didn't, you know. But the reviewer's like, no, you need to explain exactly how every molecule is protonated. Because acid wants to donate a proton, and these nitrogens all want to receive a proton. And that felt like a really tall order, and I'm not going to go into the detail for you. In case anyone's an NMR expert, you can just glance at this. But this is all the nitrogens. And you can see how on this molecule, the nitrogens are moving around. As they're getting protonated, the electron shielding is changing, and the peak is going to shift. So we had to work through it all, we figured it all out, but bottom line is that everything that can be protonated will be protonated. Now what we're trying to do here, I'm not honestly trying to make life in sulfuric acid. All my goal was to get far enough along to convince space mission sponsors that it's worth going to Venus to search for signs of life. So I'm just trying to make the case that there can be organic molecules. Now organic molecules don't mean life, but we need them for life. So my ultimate goal for my team is to make a DNA-like molecule, not our DNA, but swapping out parts. So if you were to take like the sugar molecule away and substitute with a cyclopentane, it's fine. I mean, we don't know for sure, but like that works at least for part of it. So how can we do that? Because I feel like if I can demonstrate an informational polymer is stable and concentrated sulfuric acid, I think I'd be done in terms of getting enough motivation for space missions. Well, if you've been listening to what I say and you know something about organic chemistry, what did I talk about? I said glycine is stable. Amino acids are stable. I said nucleic acid bases are stable. Once in a while, someone in the audience knows where this is headed. But based on that information, we tried PNA, peptide nucleic acid. Had anyone heard of peptide nucleic acid? Okay, at least one person or two people have. I've just put a picture here, I put its structure. But it basically has like a glycine backbone with some other things. And it already exists. It doesn't happen in nature, but people use it in laboratory work with DNA, compare with DNA, and it has the same bases. So we took that, put it in concentrated sulfuric acid, and we're able to demonstrate that a single strand of PNA is stable in concentrated sulfuric acid. We're really excited about this milestone. It breaks down at a higher temperature, but we feel like we're on a path. Now, one thing that is maybe a problem is this whole hydrogen bonding, because you know how DNA, like it pairs through hydrogen bonding, but with everything getting as protonated as possible, we're not 100% sure that it's gonna couple to itself. So we've been exploring these nucleic acid bases called alternate nucleic acid bases. This is just showing you the hydrogen bonds, but did you know that there are other bases that don't pair from hydrogen bonds? They pair from other things, like van der Waals interactions and things. And there's a list of these actually. And some people even took some of these so-called synthetic alternate base pairs and they actually put it into a life form. And of course made a company to claim they can, I don't know, make something useful out of it. But we're trying to explore too in this informational polymer we're trying to conceive of. Again, we're not gonna say, hey, this has to be what's on Venus. We just need a proof of concept to make it like a viable idea. So we have a lot of work going on here, and I just have one more example for you. And this example is of lipids in sulfuric acid. Because in the origin of life studies, people take lipids with polar heads, actually amphiphilic lipids, and they put them in sulfuric acid. And these lipids point their polar heads outwards and their hydrophobic tails inwards, and they make vesicles. And some origin of life proponents like the idea of compartmentalization first. Well, we took some, we got some colleagues and we asked them to try this in concentrated sulfuric acid. And they took some lipids, I put the ones that are there, and wow, these actually not only are stable, they form vesicles. And if you look on the right, they also have this kind of higher order behavior where they make tubules and they pearl and they split off. So it's really amazing just to see how things behave in sulfuric acid. I mean, the fact that they're stable and they have a different behavior, it's just a whole treasure trove of things. So yeah, there's a lot going on here. I wish I had more time to unpack it all for you, but we're trying to recruit people to work on this. So anyone who's interested, talk to me after. Well, what are we gonna do about all this? We want to go to Venus. We want to go to Venus and search for complex organic molecules and even possibly life itself in the clouds. But this is a tall order, and it's still too taboo for large government agencies. So we've struck off, and I formed this international consortium I call Morningstar Missions to Venus, because you see Venus in the morning sky sometimes shining brightly. And we're trying to find a way to go back to Venus or interplanetary over and over again with small focused missions, instead of waiting for what's been like 4 decades to go back down into the atmosphere of Venus. So this is going to be my afternoon talk at LPL about all the missions, because I'm not going to have time to talk to you about them now. But I wanted to give a plug for one of your professors here at the University of Arizona, because what we want to do is go to Venus with miniature molecular sensors. I want to have a probe that falls through the atmosphere with 10,000 miniature molecular sensors, each one targeted to find a certain organic molecule or class of molecules. And so there actually is one here, one idea. These tiny little, they're like 100 microns across, and they're these toroidal ring resonators actually, and she gets it coated with a certain polymer that's targeted for a specific molecule, and it can pick up molecules in the air or like in a liquid to like a picometer. It's a bit complicated to explain in the time we have. I just wanted you to know about the missions and technology, and that's another talk for later. So to summarize this part of the talk, Frontiers 2. The Venus clouds have conditions that satisfy the habitability criteria. There's a liquid, which we need for chemical reactions. Temperatures are right for covalent bonds, and there's an energy source, our sun. So the fact is, some biomolecular building blocks are stable in concentrated sulfuric acid. And sure, it's a stretch to a proposed hypothesis, but I just wanted to put it out there that concentrated sulfuric acid may be a solvent for life. Can't you just see like life on a planet with sulfuric acid, and they're studying water and they're trying to convince their audience it can be life in water? And they have to be like, no, because when you want to get a protein to fold, in some cases it has to be a very specific buffer. Too hard, too hard. Okay. So now I get to the last part of my talk, ionic liquids. And one thing I will tell you about missions to Venus is one of our concepts, we have a lot of ideas, a lot of concepts, is to go to Venus and to collect the cloud particles. Just like here when it's not happening here, right? If you go to another part of the country and it's foggy and like the fog collects on your jacket, you can have a way to collect liquid clouds and get it into a reservoir. But we'd want to find a way to evaporate the excess sulfuric acid because maybe what's in there is extremely diluted and we need to get rid of all the excess liquid to find what's left over and analyze it with, for example, a mass spectrometer. So the first step in doing that is just to generate that system in the lab, and there's my postdoc Rachna. She's now a professor in India at Indian Institute of Technology, Kanpur, and she set up just this basic pumping system. The only thing different from what you could just buy anywhere is the oil had to be like resistant to sulfuric acid in case sulfuric acid accidentally made it back into the pump. So she put some stuff in sulfuric, oh, but I forgot to mention, sulfuric acid has a very high boiling point. So you wouldn't want to boil it to get rid of the liquid because then you'll destroy the organic molecules you're looking for. So you have to put it in a low pressure system where the boiling point would be lower, okay? So that's what this is, it's a low pressure system and there's like a heater plate, she's pumping it out. So she put stuff in sulfuric acid, evaporated the excess sulfuric acid, she recovered what was in there, all good. Until one day, she went and took the amino acids or the nucleic acid bases, put them in sulfuric acid, remember it's a powder, it dissolves, evaporated the excess sulfuric acid, and there was a tiny bit of liquid left over. It's like no matter what happened, the liquid wouldn't go away. So wow, I mean, I gave you some hints. I'm like, every nitrogen wants to get protonated. We didn't know what it was, and if you're a postdoc or a professor or a student, and your job is to get rid of all the liquid and recover what you put in, but you're not. You're always getting the tiniest bit of liquid left over. Like, what's your first thought? It's not, wow, I discovered something. No. Your thought is, you made something wrong, you did something wrong. Well, fortunately, she brought it back to our very interdisciplinary team, and someone actually realized she rediscovered a substance called an ionic liquid. So remember, the acid wants to donate a proton, and these nitrogen-containing organics, they want to receive a proton at the nitrogen. So when you get rid of all the excess sulfuric acid, what are you left with? An ion, positive and negative, and they ionically bond. And she rediscovered ionic liquids. And these ionic liquids are well known in industry. There's like 1,000 of them known, probably a lot more that could be made. But they're basically salts that are liquid below 100 degrees Celsius. They're held together by ionic bonds. They're kind of like green solvents because their vapor pressure is very low. I'm sure some of you have heard of them. Even when we realized what it was, we'd forgotten we already knew about ionic liquids from a different application in aerospace propulsion. But so these ionic liquids, they're absolutely incredible. We went down a rabbit, that's the great thing about research, then you go down a rabbit hole in trying to understand what's going on. So she made a whole bunch of these. And what is so special about these ionic liquids? There's two things that are really special. Actually, there's really one thing, and that is they have a vapor pressure that in some cases is incredibly low. Like by very low, I mean like many orders of magnitude lower vapor pressure from water. And what that means essentially is they don't evaporate. I'm going to exaggerate now, they don't evaporate. So like you spill water here on this table, it's going to evaporate. You put an ionic liquid here, not going to evaporate. You put an ionic liquid in outer space, it's not going to evaporate. People are using it as space propulsion without like fully sealing their tank. Now it will get destroyed maybe by other things like high energy particles or ultraviolet, but you might consider that we could have ionic liquids on other planets where we can't have water. It could exist in like crevasses, like in rocks. It's so low vapor pressure. And it's getting to the end of my talk, and I think you can tell my level of enthusiasm, and I don't have a way to just explain to you why it feels so exciting, but I'll give you a couple things that may be relevant. A summer student did a literature review, and you can just read that bottom right corner there, but there are, unlike sulfuric acid, where something, a lot of really weird things are happening, many of these ionic liquids, it's a little more, I'm not gonna say normal, but yeah, a lot of proteins are stable. You can put DNA in some of it and it's stable. Some of them appear to remain their native folded structure. And these biomolecules are very friendly. There have been preclinical trials using some types of ionic liquids for subcutaneous drug delivery. I wouldn't put this one under your skin, of course, it's made of sulfate, but yeah. So these ionic liquids could be something really remarkable for exoplanets, for Mars, for Venus, for tons of places. And we've now kind of jump-started making ionic liquids in the lab, trying to test them. And I try to summarize it for you on this figure. This is showing you in blue a phase diagram of water. So it's log 10 in pressure of bar, but 1 bar, or 0 in log space, is what our Earth is. So our Earth is around here. We're around 300 Kelvin and 0, so we're about here. We have liquid water, but we also can have ice and vapor. But this is our vapor pressure curve. And here are a bunch of measured ionic liquids, and that's their vapor pressure curves. But people can't measure like almost 0 vapor pressure, so they just measure it at higher temperature, and you're supposed to take your eye and extrapolate downwards to what you would have at room temperature. I only found like one paper that has a special new method to measure vapor pressure at these very low, but I just wanted to show you how low the vapor pressure is. It's literally orders of magnitude lower than water. So we're trying to imagine scenarios where planets could have the planetary ingredients. What is new about this work is the ionic liquids are not made from some very rare, obscure chemicals, but from planetary materials. And any fans of Mars, you'll have heard about brines on Mars, and you hear about other bodies. So we're on this journey now to explore ionic liquids as a kind of liquid on solar system bodies, on exoplanets, and see where it takes us. So to summarize this Frontiers 3, I'm just gonna end up repeating myself because I just said all that, but they're also related to these compounds called deep eutectic solvents. Resurrection plants that you have in the desert, they kind of are like super dry, but they've made themselves into some kind of liquid that's not water. So there's a lot going on in astrobiology, and our job is really just to try to open our minds and to get away from being so terracentric and so where we are. So to summarize the 3 frontiers in the search for signs of life beyond Earth, we have exoplanets, thousands of them known. I tried to give you a bigger picture view of how we study atmospheres and how we're on the verge. We thought we were on the verge of being able to find biosignature gases. We're still on the verge, we just don't know how to close the gap, how long it will take to close our gap. I talked about Venus, and it's not my job to convert you to believe there's life in the clouds. I do hope I convinced you that some biomolecules are stable in concentrated sulfuric acid. And these are very repeatable experiments that anyone at like the senior undergrad level, beginning grad level could repeat. I briefly introduced ionic liquids, and I told you for the first time we've presented them as possible planetary liquids. So I've come to the end of my talk. I'd like to thank my sponsors, thank my team, and thank all of you for coming and for having an open mind. Thank you.
Awesome talk. I'm definitely not a space person, microbial physiologist. I'm curious about in the atmosphere of Venus, where there's all this sulfuric acid, are there other oxidation states of sulfur that you can detect without getting there, or is that unknown still?
Other than the gas, sulfur dioxide, some people think that there's H2S gas. People think, but they don't have evidence for just being solid sulfur, like chains of S up to S8. So kind of yes, we probably think that all the oxidation states are there, but can't really see them from here. Yes?
The ionic liquids, I realize that there's a lot of them and they all have different properties, but are they expected to stay in the atmosphere or precipitate to the surface?
They may precipitate to the surface. In that case, we don't know, but I'm largely talking about ionic liquids as a surface phenomena on asteroids or Mars or comets. But in terms of Venus, what's really interesting is right below the clouds, it's really hot, too hot for sulfuric acid, but there's also like unknown haze. And we're wondering if it's possible that some of those might be ionic liquids after the ionic liquid evaporated. There's a small chance. And one of our missions that's going to Venus could weigh in on that with some evidence. So they're very heavy though. They tend to be pretty dense. And they're pretty gel-like. So my guess is they probably precipitate out and that the surface is the best place for them.
I have another question.
By the way, I forgot you, that reminded me. I brought a little bit of ionic liquid to show you, but they're all very different, so they're not all this viscous. So I will start passing it to the students and we'll see how far it gets. Okay, yes?
And is there a connection between the degradation of your diatomize and the formation of ionic liquids? Is what we're seeing in those stable
Yeah, that's a good question. Not in this case. Not in this case because in the NMR we see that the dipeptide is still there, but the NMI for the ionic liquid shows something else. And in that case, the dipeptide is just a tiny amount in a lot of ionic liquid, or in a lot of sulfuric acid, but the ionic liquid's like a 1-to-1. So we don't think it's forming with such like a dilute amount of dipeptide. Yeah. Yes?
Can you expand a little bit about the research? So you are going to do missions?
Yeah, so I'll preface it by saying you have to be ambitious. You never know what's going to happen, whether these missions will get done. But we came up with like a series of 3 missions. And this was all motivated by the phosphine on Venus, because we can go to Venus and see what's there. And so we have like small, medium, and large, short term, medium, longer term. And our first mission to Venus, we call it the Rocket Lab mission to Venus. And that mission is pretty much a go. We have an instrument that exists, we have a flight spare, we put it on a drone and tested it. We have a probe, we've got a pressure vessel from Rocket Lab. We're still a ways off from actually launching. And this first mission is pretty straightforward. It's like throwing a rocket at Venus. I shouldn't really say that online because it's not specifically true, but very simple mission, single instrument, that's 1 kilogram. We're gonna shine a laser out into the clouds, have it bounce back, measure the backscattered polarized radiation. We're gonna look for fluorescence. So that's the first mission. But the second mission, we wanna send a parachute mission to Venus. And the parachute would, I call this my baby I gave birth to this summer, this idea, but this mission would have a parachute that's 5 meters in diameter. It would have like almost a bowl on the bottom of a probe attached to the bottom with these patches, and each patch would have hundreds of miniature molecular sensors targeted for different molecules. And we're exploring a lot of them. Carbon nanotubes, this UV decay thing. There are these chip-based frequency cones, which probably won't work out. There's the Jujitsu one. So we really want to do this, and I'm going to expand more on this in my next talk, so I don't want to, I know everyone doesn't have time to do the other talk, so I'll just tell you one fun thing. There's 100 steps from idea to execution. So this summer, we wanted to test this idea out, but we couldn't send 10,000 miniature molecular sensors to Venus, and we don't have the sensors yet, so we did an Earth-based project. We put 32 temperature, pressure, humidity sensors, and this was an undergraduate project, and this is a master's student who designed a parachute that can be scaled to go to Venus, and it's based on the design of a parachute that already went to Venus. And just to make a really long story short, we launched with a weather balloon, and we went up to, not space, that's her parachute, there's our miniature sensors. We got a photo at 70,000 feet, and we have a platform now to go back and test the miniature molecular sensors next summer, whatever's ready. Yeah. Yes?
Can you comment more on the PNA and whether you think that can be naturally occurring?
Good question. I can't comment on whether it would be naturally occurring. I know I cannot. I can say that we know from Bennu, an asteroid that NASA brought a sample back from, they found all the nucleic acid bases and like purine and other things. We know amino acids are out there, but I don't know if it formed naturally. Maybe it did. Maybe it formed here on Earth at one point naturally. It's speculation, but good question though. Yes?
So I was wondering, I have kind of 2 questions. One of them is more of a clarification. So when you were testing the biological materials and the ionic liquids, were you testing just the amino acids, or were you testing everything you talked about?
Good question. The review I showed you was a literature review where other people did it in other ionic liquids, not the one I was talking about. We have been able to put some DNA. It's been a great adventure. My postdoc Aero, he's doing cryo-EM. Any of you got to try that? That's like super hard. We put some molecules in, but we're trying to do everything we've done in sulfuric acid in all the different ionic liquids that are planetary based. So that is work TBD. It's not what we've done yet. Yes?
Have you tried combining these organic molecules, like the lipids and the peptides of the ionic liquids in the sulfuric environment?
Great question. Not yet. No. But I love that. Yes, we want to try to do the next set of things. And to that point, I want to say what just started out to motivate missions to Venus or to help get money to go to Venus, it took a whole life of its own now. So we want to do all those things. Yes. Yes?
In regards to also the idea of getting the sulfuric clouds to boil using that pressure change, is there any thoughts of how anything else that you're hoping to find in the sulfuric might also react to the pressure change? Or is it just thought that those will mostly be fine while the sulfuric acid boils away?
I think they'll be fine. I think you can do the experiment on Earth in the lab and show that the molecules will be fine. It's not a super low pressure anyway. We're not going to like ultra high vacuum. It could be like Mars's atmosphere or a bit less than what Mars has. So it will be fine. Yes?
So what in an atmosphere with way more sulfur, which amino acids would you expect? I'd expect a very different set to the current 20. And I know, there are others we think were early on Earth, like ornithine or leucine or valine, whatever. Have you tried a broader range of amino acids and ones that also have more sulfur in them?
We haven't. We're recruiting though, so maybe you can try it. No, I'm just saying. We definitely want other people to do it, and we don't have enough time. We're like, a tiny team, like on a bit of a shoestring budget, but definitely, we just want to promote the amino acid backbone is stable, and I'm sure a lot of these other ones, especially if they're sulfonated or sulfated to begin with, will be stable.
So you tested like the nucleic acid base is pretty stable, but like what about the nucleotide? Just like a little bit harder.
Yeah, the nucleotide is not, because sugar is not stable, and also the sugar phosphate backbone is unstable. But we did put, and we have a drug company, by the way, that we found that we work with to synthesize the molecules, because we can't get that far. And they've been really great. So we did ask them to do like a cyclopentane on a nucleic acid base. And cyclopentane itself is not soluble in sulfuric acid, but attached to the nucleic acid base it is. So we did that. But then we shelved that when we thought of PMA. So yeah, the nucleotide is not stable.
No more questions? Thanks, everyone.